Okay, it looks like numbers are leveling off here, so why don't we get started? This is part 3 of the Working with eBird Status and Trends Data Products webinar. And today we're going to be talking specifically about trends applications. And just a reminder that we've already had 2 of these webinars, which I imagine most of you have attended the past 2 weeks. The first one was an introduction, then we did applications of the status data products, and then today we'll talk about trends applications. And the previous 2 webinars are already on YouTube if you want to watch them, and then this one will get up on YouTube within the next few days. The general kind of overview for today, I'm going to start by recapping a little bit of the stuff from the previous webinar. I'll take up the 2 exercises that the homework exercises that I left you with. That should only take a few minutes, and then we're gonna dive into the trend stuff, and Tom will talk about the trends methodology. And then following that, I'll do some R examples for some basic applications of the trends data, and then we'll go to questions. Um, I realized that just now that there may be a few people here who weren't in the other 2 webinars, so maybe I'll just actually introduce myself. I'm Matt Stramis-Mackey. I'm a data scientist on the status and trends team here at Cornell, and yeah, maybe Tom and Andrew, you guys want to give a brief intro for people who haven't been here?
My name's Tom Auer. I'm a lead geospatial data scientist in the Center for Avian Population Studies and help lead the status and trends production.
My name is Andrew Stillman. I'm a applied quantitative ecologist here at the Cornell Lab of Ornithology, working on the conservation team. My job is to take the outputs of status and trends modeling frameworks and make them really useful for bird conservation. And I work across a number of different wildlife agencies here in the US.
Okay. So let's start with those homework exercises I just talked about. Okay, can you all see my R screen here? Good. Okay, I'm going to make it slightly larger for folks. All right. So this I'm going to take up the 2 exercises we covered. I gave to you all at the end of last session. This is all in basically the same R script as I've been using for all 3 webinars. So if you already have the link, it's the same link. And I guess maybe Andrew or Tom, you could just repost that link for people that just happen to not have it. But today we're going to start around line 263. So the first exercise was to recreate these migration chronologies, but this time, rather than show the proportion of population throughout the year, the portion of the entire global population throughout the year, make a slight modification and just show the proportion of the US population throughout the year. So this is, I'll just step through the code briefly. On the right here, I have the bottom right, I have the figure from the global population case that we did during the workshop. And then we'll see how that changes when we adjust it to be the portion of the US population. So it's the same set of grassland species that we worked with before, and this is just loading the Montana boundary, also that we worked with before. But now the new piece is that I'm also going to load a boundary of the US. And just for this example, I'm doing just the contiguous US, so the US minus Hawaii and Alaska. And then I have the same, essentially the same loop here that goes through, downloads the data, calculates the proportion of population within Montana, as well as the upper and lower confidence intervals of that proportion. And it does that for every week and for every species in this list of grassland species. The only modification here is this bit right here that I've highlighted. So when I load the relative abundance raster, the first thing I do is I just mask it to the US boundary. So that's just going to remove any of the values outside of the US. And then everything else is essentially the same. So I'll just rerun that. As that runs, I'll just say that for those of you who did not attend the previous session, this may be a bit confusing, but you can go back and watch the YouTube video to get the full details on exactly what we did in that session. So it's just looping through the species, calculating these proportions of population. Looks like it's done. And then I have some ggplot code here that will make the updated version of this plot on the bottom right. Oh, I screwed something up. What did I do? One second. Just going to actually restart my R session and see if what happened here. All right, let's try this one more time.
Matt, while you're running that, we have someone asking that the mask only removes the values outside the USA, but does not recalculate the values.
No, yeah, it's so the rasters that I'm loading are relative abundance rasters. And it's just gonna, anything that's outside of that US polygon, it's just gonna convert those to NAs. It's just gonna remove them, but it's not gonna change the values inside the US. But after I load the relative abundance data, I divide it. I divide the relative abundance by the totals, the sum across the whole region. And now that total is different because it's not the global total relative abundance, it's just the relative abundance within the US. So the proportion of population raster will be different because it's now each cell is the proportion of the population within the contiguous United States. Okay. Let's see if this works now. Okay. I have a sneaking suspicion this may still be problematic. No, okay, that worked. That's like the number one trick for our problems is just restart the whole thing and like and try again. Like that works like 90% of the time. Okay, so this looks quite similar to the previous one. Basically the main difference is that the proportions are now larger because we've removed, we're ignoring the proportion of the proportions of the population that are, for example, in Canada or Mexico. So that's a tiny little modification you can make to these to make them more tailored to your region. And that could be, you could, if you're interested, for example, in like the proportion of the whole North American population, you could equally do that. I'm going to do the next exercise, and then I'm going to talk sort of a bit more generally about how these exercises could be applied like in the real world. Okay, so that was an exercise kind of looking at the temporal side of the data, and the next one we'll look at the spatial side of the data. If you remember in in the workshop, we generated these maps that show areas of importance for a set of species. And the exercise was to choose a different set of species in a different area, and make a similar map. So I've chosen this set of 6 woodpeckers as an example, and I'm going to do the example for Washington, which is the state that I live in. And I'll just note right now that I've intentionally chosen some migrants and some residents, because there was a question using exactly the set of species that I got after the workshop about how you handle migrants and residents. So let's load the set of species. Let's load that boundary for Washington State. Now, again, I have a for loop. I'm going to loop over the 6 species. The only difference in this case is that in this stage where I'm loading the seasonal, I load the seasonal proportion of population rasters. If you remember from the case where we had all migrants, we loaded that raster and then we subset it to the breeding season. But residents don't have a breeding season. Residents is just we have the whole year. So all I've done is I've added this little if statement, which says, if the species is a resident, instead of getting the breeding season layer, we'll get the resident layer, which is another name for like the full year layer. But otherwise, it's basically the same. We're just swapping in the resident layer rather than the breeding season layer. So that was taking these proportional population rasters, masking and cropping them to the region of interest, which is Washington. And now I'm going to take the mean across those 6 species. I'll remove the zeros because these are areas where none of the species occurs. And then I've arbitrarily picked the 70th quantile as kind of a cutoff. So anything below the 70th quantile, I'll remove to highlight just the 30% of most important cells for the set of species. And then I'm just going to make a map. Okay. So this is a map that shows you areas of importance for this 6 set of woodpeckers in Washington State, where purple are the lowest importance areas, and then the yellows are the highest importance areas. So that was super quick. I just wanted to kind of take those up live. All the code is available to you in the R script, and then, as I said, the details on exactly sort of going through it more slowly will be in the previous webinar's YouTube video. But I did just want to take a couple seconds because we got an interesting question in the Google form after the second webinar from someone who said they work in a field where they give conservation recommendations to farmers, and they were particularly interested in understanding how these 2 examples could help them identify critical times and critical places for conservation interventions or conservation recommendations to these farmers. And this is like a really common, this is sort of what makes status and trends data products so valuable, is that, there's a million things you could do for conservation, but like your resources are always constrained. So it's critical to be as strategic as possible with your conservation resources, with your conservation investments. And part of that is really precisely directing those resources to be in the right place and at the right time. And these 2 examples were representations of that. This map here is an example of in the right place. It tells you what are the most important places for this set of woodpecker species. And the previous example, maybe I can just put these chronologies, is an example of at the right time. I was hoping it would pop that up again, but that's fine. They tell you specifically for a given region, when are a set of birds that are of importance to you in that region, and when are they there at the highest abundance. So that can tell you specifically when to apply some certain conservation intervention. And I won't go into it in too much detail, but I did just want to post in the chat. There's a really cool example from a couple years ago, or I guess a few years ago now, that the Nature Conservancy did using this kind of targeted in space and time conservation intervention in the Central Valley of California. So I'm going to post a link to just a simple, it's not a scientific paper or anything, it's just an article about that project. It's called Bird Returns, where they had farmers leave water in their fields to make them better habitat for shorebirds, but they had them do that at very specific times of the year as the shorebirds were just briefly passing through these areas on migration. Okay, so before we jump into trends, I just wanted to ask if Andrew especially had any additional comments on that type of application stuff that he wants to add. All good? Okay, great. And were there any other things from the chat that we should address, or can we move on? All good?
I think we're going to move on.
Cool. Next up, Tom's going to talk about the trends methods.
Okay, thanks, Matt. Yeah, so if you saw the Birds of the World webinar last fall that I did, I gave a very brief overview of how we put the population trends together at a very high level. And so today I wanted to kind of step down just one level more detailed and give a slightly more nuanced explanation of how we generate the trends results so that people have a little bit of an understanding of how they're made and what they are. And then Matt will jump into applications. So our goal with this is slightly different than with the status products. With this, we want to we also want to be spatially explicit, so we want to capture spatial variation, but we want to understand trends in population over time. And there's basically two key challenges that we want to address in this situation. And the first and most important one is confounding. I think anybody who's aware of, eBird as a participatory science project is aware that people change what they do over time, simply, based in terms of like where people are going and the program growing at all, right? More people could mean more birds. That's the confounding and the variation over time that we need to deal with. So that's the first major challenge. And then the other one is that trends are happening at different scales locally and regionally, and that changes across species range. And so we want to capture that spatial variation, which is kind of unique to some of the trends modeling that's been done in this realm before. So I'm going to give some kind of simplified versions of the trend models and try to just explain a little bit what's going on with what we're doing. So we've talked about status before, and status is essentially a species distribution model. We use machine learning and random forest, but we have a set of features and we predict where birds are. And we do this for 1 year, the most recent year. And we try to do it for 52 weeks of the year. And the outcome that we're looking to predict, to estimate, is relative abundance. And we do this with these SDMs, species distribution models, using a suite of habitat and effort covariates. And so if we want to extend this then to understanding the change over time, we want to develop a trend model. And conceptually, maybe it's not all that different what we would like to do. We'd like to get yearly relative abundance. So we have an equation in our model that says take the SDM and add a variable for change, right? What's going on year to year. And so that's kind of the conceptual idea, is that we want to understand what's happening at a yearly basis, and the modeling method wants to calculate a percent per year change in relative abundance. And so that's the goal, but as I kind of set up, confounding is the problem. And so this is an example of checklist density on a log scale for the Eastern US from 2007 through 2018. And the simple growth of the program is a confounding factor, right? Where there are more eBird checklists over time, is changing over years. And this could be confounded with changes in birds. Similarly, what time of day people are going out to survey birds has changed over time. So this is a density plot showing what time of day participants are going out as the program has changed. So each line is a different year. Red and orange, yellow are early parts of the eBird program. Purple, blue, red are later in the program. And you can see that early on, people were primarily going out first thing in the morning. But as the program grew and it had a broader audience, it kind of started to conform to more usual human activity hours, with some preference still towards the morning, as that's when birds are most active. So this sort of change through time is another source of confounding. And then there's a variety of other things going on with how people are learning, how knowledge is being shared. You can think of field guides or apps like Merlin, social media where information about where birds are spreads. And things like COVID actually drove people to choose a different type of habitat, right? The proportion of urban habitat in where people were going trended up in California when there was a strong lockdown, and people mostly birded where they were living, not going to parks and things like that. So that sort of thing can confound what's going on with birds, because those are things that are going on with the observers in the observation process. So this method that I'm going to kind of preview and talk about, the goal is to try to deal with this basically selection bias problem through time. And the primary method that is used to do this is to construct what's called a propensity score. So I've got that equation up of our goal here where we want to estimate yearly relative abundance. So we're going to take an SDM and add a variable for yearly change. But the problem is there's this kind of factor confounding that is causing a problem. It's affecting our ability to estimate that yearly change. And so the goal is to try to correct for that. And this comes from causal learning and inference fields in economics and statistics. And so we're using that framework of causal learning to basically understand a treatment. What is the likelihood that a checklist is from a given year? What's the probability of the checklist year? And this is our propensity score model. So it takes a lot of the same features that we have, but instead of estimating relative abundance or yearly relative abundance, this propensity score model is estimating the year that the checklist came from. And so that gives us a way to understand what the bias over time potentially is in this causal learning framework. So this value, these propensity score models, then go in to the next model, which is where the paper about this comes from, which is double machine learning. Daniel Fink led a paper that was published in Methods in Ecology and Evolution last year describing this double machine learning. So the double part is, first we calculate this propensity score that estimates the year of the checklist. And then that goes in as a factor in the trends model. So now we have a trends model that is estimating relative abundance between two time points, and it has that change factor in it. And then it's taking an adjustment. It's using that propensity score to control for those confounding changes over time in the model. And the one other thing I'd just like to point out here is that two other things I'd like to point out. One is that the variables that go into the trends model itself are going to differ a little bit. We really only want to put features into that trends model that are capturing changes over time in what's happening with the birds. So land cover, for example, or things like that. And then finally, you know, having those habitat features, those yearly varying habitat features in the trends model, that's what lets us capture spatial variation in the trends of the birds themselves, while trying to use the propensity score to remove as much of the confounding bias as possible. And we have a number of features that do that in the models, that the model can learn what's going on with that spatial variation, and some of those are listed here. So that's the conceptual framework and kind of some simplified modeling equations for what we're trying to do. I'm just going to show you a little bit now how we kind of implement this and validate it. And so we do this with a simulation study. And the goal is to basically simulate data that is replicating where birds are, their abundance, the eBird observation process, and the confounding in the eBird data. But we're going to apply known trends to this simulated data, and that's what the value of the simulation is. And we do this for every species we model. And we're going to walk through a couple examples here. One is wood thrush, which has lots of data and is fairly prevalent. And then we're going to talk about long-billed curlew, which has both less data overall and is less prevalent during its breeding season. And we're going to use this double machine learning implementation, which uses a hurdle model very similar to the status one in a sense. It has occurrence and count conditional on occurrence. These are all random forests. They're from the GRF package. And then we're going to do a lot of sampling. We're going to have 100 replicates, and Matt will touch on how you use these later, but that replication gives us some uncertainty information about our trends as well. So let's look at these constructed known trends to get a sense of what the simulation is doing. So we're going to start with spatially constant trends. So on the right here is that simulated data I was talking about that is trying to capture what's going on with the bird, what's going on with eBird observation process and confounding. And in this constructed known trend in birds over time, we have a spatially constant, consistently declining trend baked into that simulated data. And so we've simulated that data process, and now we're going to take that simulated data and run it through that trends model that I described with all those parts and see how well does it capture this simulated pattern. And so on the left here, you can see that range-wide, it's doing a very good job of that. There are some places where maybe the magnitude varies from the simulated data a little bit. But by and large, we have captured both the direction decreasing and magnitude more or less moderately decreasing range-wide trends in the species. So when we do this, we feel confident that what we do when we run the real eBird data through the model is going to give us a result we have some confidence in. Similarly, we do this for spatially varying trends, as we all know that species, as I've talked about, have spatial variation in both where they are and what the trends and different parts of their range are. So on the right, you can see a simulated trend related to forest cover, decreasing in those forest covers, increasing outside of that. And on the left, you can see the estimate from the, the trends model recovering that signal and that spatial pattern very nicely. These next slides, you don't have to pay attention to all the details, but the point is that we do this a bunch. We have a variety of signals. We have spatially constant, spatially varying. They range from weak, moderate to strong. And then we do the replication of this. And what it gives us is basically this scatter plot of simulation values and estimates of those simulation values. And we can look at the correlation between the two and know whether the model is likely to perform well on the real data. So wood thrush, really high, really well correlated. You can see that the patterns between these pairs on the map match pretty well. For long-billed curlew, where we have less data, there's a little more noise. The correlation is still high, but the width of that band of data is a little bit wider. And if you were to focus in on some of these pairs, a couple that are highlighted here in green, if you look at them left and right, there are more substantial differences between the two simulated and estimates and how they're varying. But still at an 88% correlation, fairly confident that what we're generating in terms of real data estimates of the trends are reliable. And so just to wrap that up, this method, this double machine learning, is trying to capture spatial variation patterns while controlling for confounding in the observation process with eBird data. I've linked to the paper there, which you can also find on our status and trends FAQ. And if you go to the status and trends page, you can view these trends there.
There's a question in the chat.
Yep.
For you, Tom. Does this analysis assume no correlation between confounding and trends?
I don't know if no correlation is fair. I think, this method is doing a reasonable job at controlling for as much confounding as we think we can, but it's not a 100% confounding control.
But you're removing all known sources of confounding
Right.
that we can actually model out.
That we can model out, yeah. If there are unknown sources of confounding that we either as analysts and bird people don't know, then that may still be a residual in the estimates.
New question for you, Tom.
Mm-hmm.
Is the same data used to train the SDM and propensity model? If so, is there any risk of bias there?
So it's basically, there's two different sets, and I see, I don't think I have a slide on that. No, I don't have a slide on that. Basically, they are similar but different sets of data. We don't include things like basically a bunch of the effort information and a bunch of some of the like fine resolution stuff like weather data or things like that in those models. It's generally seeming to be not good to have some of the interannual varying stuff in the propensity model as you do in the trend model. So in the trend model, we're really focused on putting the habitat features that are going to be relevant to the changes in the birds so that we can understand that spatial variation.
Next question. I'm not sure people can hear me that well, so if you could repeat the question.
Sure. Yep.
Given that the model is estimating trend between 2 time points, what happens if there is a temporal dynamic of, let's say, initial increase, stationary distribution, no change, and then decline? The trajectories.
The trajectories, yeah. So as soon as I turned on my slides, I said, I wish I had a slide showing kind of the conceptual of what we are estimating. And we're estimating 2 time points, which I think the question is about what happens if, as you're estimating the change between a start year and an end year, there's a lot of difference between those 2 time points. Like there might be a strong increase and then a stabilization. Yes, if there are strong year to year variations within that 10 to 12 year period, we are not, this is not describing it. We are calculating an average effect over that 12-year period. So if there is some of that interannual variation and it has an overall effect on the start and end point, it's somewhat going to be captured, but we're not generating estimates for each specific year in that whole time period, which we've come to call a trends trajectory. That is a focus of research right now. All set? Great. All right, I'll stop sharing and pass it back to Matt for applications.
Okay. All right. So actually, but before we get into the R stuff, I thought I would give a little bit of context. We're going to use, for examples, some species from sagebrush habitat in the Western U.S., but I think there's some people on the Zoom that aren't familiar with these species. So I'll just very quickly show you a couple things in my browser. Oops. Are you guys seeing the sagebrush photo?
Yep.
Okay. This is, I just Googled photos of sagebrush. So this just gives you a sense for what the habitat looks like. And it's one of my favorite habitats in the Western US. And one of the species we're going to focus on is this sage thrasher, which breeds pretty much exclusively in sagebrush in the Western US. And then the final little bit of context, I'm going to use, rather than states, which we've used as region examples from the previous webinars, I thought I would switch things up and use a case where we actually have to download a polygon from the web. So I'm going to use bird conservation regions. And these are areas that have sort of common sets of birds and a common set of habitat. They're defined within North America. And I will actually just post a link so you can look at this map if you're interested. We're going to be, as an example, I'm going to be using BCR 9, which is called Great Basin, I believe. Okay. So let's get into the R stuff. Oops, I shared the wrong window. Let's try this one instead. Sorry, I have multiple R sessions open. That one? Oh, it keeps going the wrong one. I'm sorry. There we go. Okay. I think now we're up and running. All right. Just gonna restart my R session. This is the carrying on from the same R script as before. I'm gonna load the packages right at the top. The only new package today is this one geodata. And we'll see shortly why I have that. And then I'm going to jump down to the section for the trend stuff from today. Okay, so far we've only talked about the status data products. The trends data products they're downloaded and you work with them using the exact same R package, that eBird ST R package. And I'll show you today just how you can do it. In a lot of ways, they're actually easier to work with than the status data products. So this section should go pretty smoothly, I hope. So if you remember from, I guess it would have been the first webinar, the introduction, I showed you this eBird ST _runs data frame. And it's a data frame that shows you a full list of all the species for which we have eBird status and trends data products. Now, on the trend side, only a subset of that full list of species actually has trends. And if we want to subset this data frame to just those species that have trends, I can filter the data frame. There's a column in this data frame called has trends. So I'm going to filter it to just species that have trends. And then let's take a look at this data frame. So the first part on the leftmost columns on this data frame, you should already be familiar with from parts 1 and 2. So you get like the common name and scientific name, and then a bunch of information about the status, like the season dates and so on. But if we scroll all the way over to the right side, these remaining columns are now all related to trends. So if you recall from part 1, when we show the trends maps, trends are only calculated for one season. It's not like status where we do it for every week, we just do it for a single season. And this column here called trend season, tells you whether it's the breeding season, the non-breeding season, or whether it's a resident, in which case it's a trend for like essentially the whole year. Also, trends is not calculated for necessarily the entire global range of a species. It's calculated for a region. Most species are have trends calculated for North America, but we have some species, for example, this one, which we have trends for India and Southeast Asia. Then we also have the start and end year of the trends. Those years are consistent across regions. So all your North American trends are going to be 2012 to 2022. And then you have the start and end date, the start and end dates, the dates that define the season. So this was this first line here. It's a North American trend, and the dates are from the 25th of January to the 10th of May. So that defines the period over which the trend is being calculated. And then finally, we have some metrics that tell you something about the quality of the results. These are measures that compare, that are based on the simulations that Tom talked about, and they tell you how close our estimates match the true known simulated trends. But I won't go into that too much today. Okay. So that gives you a sense that would be how you could find what species have trends. And in that data frame, one of the species is sage thrasher. That's the species that I just showed a photo of a second ago. So if you want to download data for sage thrasher, it's very similar to downloading data on the status side. The only difference is instead of eBirdST download status, you use a function called eBirdST download trends, and we just give it the name of the species. Unlike on status, where there's dozens of different data products available, trends, there's just one or two files that get downloaded, so we don't have all this specificity to be able to pick out certain files. It's sort of like you just download the data, and that's it. So I misspelled sage thrasher. So that will download the data. This is assuming, we talked about in part one, that you already have an access key set. If you don't have one, you can go back to the first webinar. I already had data downloaded for the species, so it's just saying that I already had the data. And then if you want to load the data into your R session, the function is load trends. And again, give it the name of the species. Now, unlike on the status side where we were working with GeoTIFFs, on the trend side, this is just a simple data frame. So we can look at it. Actually, I'll look at it this way. Oops. So it's just a data frame, not some sort of fancy spatial format. The first several columns are exactly the same as the columns in the eBirdST runs data frame. So it just tells you the species, the season, the start and end year, the start and end date that define the breeding season. And then this SRD ID, these trends are calculated on a regular 27 by 27 kilometer grid. And the SRD ID is just a unique integer that defines each grid cell. And then to the right of that, you'll see a longitude and a latitude, and that gives you the coordinates of the center of each grid cell. And then you have the trends themselves. So first, there's actually a relative abundance value, and we'll see why this is really valuable shortly. This relative abundance value is actually the relative abundance of the middle point of the trends time period. So this is a trend from 2012 to 2022. This is a relative abundance value for whatever the midpoint of that is, like 2017. Then we have the percent per year trend in relative abundance. This is expressed as a percent. So minus 1.57, that's minus 1.57% per year over that 2012 to 2022 time period. Then we have 80% confidence intervals around that value. This true false value just tells you if it's true, it means that the confidence intervals don't overlap 0. So we're fairly confident at least in the direction of the trend. And then at the far right side, you have another set of values that's the trend expressed not as the percent per year, but as the cumulative trend over the full 10-year time period. And when you look at the Status and Trends website, you look at the interactive maps with the, you know, the blue and the red dots. What's expressed there is actually this value. It's the cumulative trend, not the percent per year trend. But the models fundamentally calculate percent per year trend, and then we just convert it to cumulative. So here you have both. Okay. So before we get into some applications, I thought I would show you, oftentimes folks want to basically take this tabular form of the data, convert it to some spatial form, and then pull it into like ArcGIS or QGIS. And it's pretty easy to do that. And we actually have to convert to a raster format. We actually have a function in the eBirdST package that will basically do that for you. So let's say I want to convert the percent per year trends into a raster format. The function is called rasterize trends, and I give it that data frame. It understands the structure of that data frame, and it has information about the location of all the cells and the shape of all the cells, and it knows how to create a raster based on that. So after I run that, now we have a raster. This is, for those of you that attended the previous two sessions, this should be pretty familiar. This is the same kind of format. It's a spatial raster format with 3 layers for the median and then the confidence limits of the percent per year trend. If you want to save that to your computer to then open it in like ArcGIS or something like that, you can just use the write raster function, which is from the Terra package. And we'll give it a name. I'll say Sage Thrasher abundance percent per year, 2012 to 2022. And then .tiff will tell it to save it as a geotiff. So you can save that and then open it in ArcGIS or QGIS. The other way you could convert this to a spatial data set is converting it rather than to a raster, and convert it to a set of points for each defined at the centers of the raster cells. So to do that, I'm going to use functions from the SF package. I'm going to convert it to an SF object. All I have to do is define what the coordinates are for this data frame, in this data frame, the columns corresponding to the latitude and longitude. And I have to tell it the coordinate reference system. A short form for lat long, like unprojected coordinates, is the code 4326. And then if I want to save this to my computer to open it in a different software, this time I'm going to use the function write_sf. I'll give it the data frame or the SF object, the spatial object. I'm going to call this one Sage Thrasher trends 2012, 2022. I'm going to save it as a geo package, which is sort of like a more modern, fully featured form of a shapefile, but you could also save it to a shapefile. And I'm going to give it the layer Sage Thrasher trends. So that'll save it to my desktop or to my computer. The last thing I want to do before I get into the applications, there may be situations where you want to convert between the percent per year trend and the cumulative trend. And I just wanted to put this formula in here so you'll have it in case you want to do that.
Matt, can you just like elaborate on that just like a little bit more, the difference between the two? We had a question about what.
So you can think of this as like it's basically the exact same as like compound interest. So it's like, oh, say you have, you know, $1,000 in your savings account, and that that account gives you 1% interest per year, then you can, there's a formula that you can use to then take that per year interest rate and successively apply it so that you can say that, oh, if I have $1,000 now and it's a 1% interest rate, then after 10 years, then I have, 1,000 plus whatever. So it's the exact same thing, but with birds, so it's like, if you have 1,000 sage thrashers, and in this example I have here, I'm saying PPY trend is 2. So that's like, there's a 2% increase in sage thrashers per year. And saying I'm starting in 2012 with 1,000 birds, and I want to know how many birds I'll have in 2022. This is the formula. So you end up, so this isn't the number of birds. This is actually the percent change. So basically, like, after that 10-year period, the population will have increased by 21%. So for example, if you knew that you had 1,000 birds, you could then multiply it by like the cumulative trend, and it would give you, oh, sorry. I have to then like divide this by 100 or something, and then add 1. Oops. So hopefully that makes a little more sense. And just to be 100% clear, like internally, like when we do this modeling process, it's always percent per year. That's what comes out of the model. But when we show it on the website, we show the cumulative trend. So, it will be significantly larger because it's a cumulative number over 10 years. And in the R package, when you download the data, you get both. So it's totally dependent on what your application is. Sometimes you may want to work with cumulative trend. Sometimes you may want to work with percent per year trend.
Matt, quick question about the raster metadata of SRD long lat. Is that available outside of load trends or no?
Oh, like if you wanted to just know the coordinates of every cell with basically no. You would have the only place it's currently available is if you actually load the data. We could potentially add that kind of like a function somehow that would just give you a list of all the cell IDs and the lat and long, but we don't currently have that. Okay, good to move on. All right. So I have three examples. The first one's really short. So let's get into that. So kind of the most common use of the trend is there's some area that's of interest to me and some bird that's of interest to me. And I want to know what's the proportion what's the trend within that area. So for this example, I'm going to work with those bird conservation regions, and I'm going to work with BCR number 9, which is I think it's Great Basin. This is basically designed to show you how instead of getting a state using an R package, you could just load a shapefile from your computer. Okay, so let's say BCR equals, you use the function readsf to read a shapefile off your computer. I downloaded already those BCR polygons, but you could download them yourself from that link that I posted in the chat. And they're in a folder called BCR terrestrial. And then the shapefile that I'm interested in is BCR terrestrial master.shp. So that's going to load the shapefile into R, and that's going to that shapefile has polygons for all the BCRs. I'm only interested in BCR number 9, so I'm going to filter that to just BCR 9. There's a column called BCR, which is the numeric value of each of the BCRs. And then the trends data are in this lat long unprojected coordinate system. So I'm going to transform this data set this set of polygons to that same coordinate system. So I'm going to use ST transform and give it, remember, it's that same 4326. That's the short form for unprojected lat long coordinates. Okay, so it's just a polygon defining the boundary of that BCR, the Great Basin BCR. Next, I want to load the trends for Sage Thrasher. We already did this before. I want to convert them to a spatial format. We already did this before as well. So I'll just ST as SF, trends, coordinates, these are the longitude, the names of the columns in the data frame specifying the longitude and latitude, and I give it the coordinate reference system. Next, I now have a set of points for the whole breeding range of Sage Thrasher that each point gives me the percent per year trend at that point. I just want to subset that to only the points that fall within BCR 9. There's a variety of ways you can do this, but I think the most intuitive is you can use the sort of standard square bracket subset notation, and it also applies spatially. So this is sort of short form for give me the points in trends SF that fall within the BCR polygon. So what I have is a set of 900 locations that fall within that BCR polygon. And each location has a trend associated with it. So the sort of naive thing you might do here as well, I have 900 points. I know the trend for each. Let's just take the average across that. That doesn't account for the fact that some locations, some of those 27 kilometer cells may have many more Sage Thrashers than others. What we want is the population, the sort of average population trend across the whole BCR. So we have to weight the trend by the abundance. Rather than just taking a straight mean, we're going to take a weighted mean. And that's exactly why we include, when you call that load trends function, it's going to give you the abundance as well as the trend for exactly that purpose. So you can do this sort of weighting. So BCR trend is just sort of a standard weighted average, where I'm going to take the abundance at each cell and then multiply it by the percent per year trend. That's the numerator. And then I'm going to divide by the sum of the abundance across all the cells. Okay, so what this tells us is that there's a minus 2.4% per year trend for Sage Thrasher within BCR 9. The next application is going to just expand that one step further. And it's complicated in two ways. Number one, it's often really important to have some sense of the uncertainty involved in that trend. So how confident are we in that minus 2.4%? And we have a way of calculating the uncertainty of that. And also, I'm going to modify a little bit to calculate not just a trend for a single polygon. I'm going to do it for all the states in the US at the same time. And there was another question from last time. Someone was interested in how you use this GATM data set rather than the natural earth data set to get boundaries of regions. So in this example, I'm using that. There's a function in the geodata package called GATM, G-A-D-M, that allows you to access country or state polygons from that data source. So in this case, I'm saying country equals USA. Level equals 1 means 1 is states, whereas 0 would be countries. And then again, just for the sake of mapping, I'm excluding the Alaska and Hawaii. Because if I make a map including Hawaii, it's gonna be really hard to see the core part of the range. So you see it downloaded the data set and then loaded into my R session, and now I just have a set of state polygons for all the states in the US. Okay, so to calculate the uncertainty on the trend, you'll recall Tom mentioned in his portion that we have these multiple folds, multiple estimates of the trend at each cell, at each 27 by 27 kilometer cell. In fact, we have 100 estimates, 100 trend estimates at each cell. And in the R package, we actually provide access to that data. So it's a different data frame than the one that we've already accessed, which shows that the values are summarized across those 100 folds. If we want to load the fold level trends, the function is called, it's the same load trends function. I'll give it the name of the species as always. But now we say fold estimates equals true. So I'll do that and we'll look at this data frame. So it's a simpler data frame. There's not as many columns. This one just has the species, the season, that same cell ID, latitude and longitude. But now you'll see here there's a column for fold. And every cell will have 100 folds. And corresponding, there's 100 different abundance PPY, percent per year abundance values. Okay. I want to, just as before, I want to convert that to a spatial format because I want to assign the cells to the states. So trend, I'm going to call this one trends folds SF. I use SF because SF is a package that we work with point spatial data. This is identical to what we did before. It's just using the same, we're specifying the latitude and longitude columns and specifying the coordinate reference system. The next step is I want to take this spatialized set of fold level trend estimates, and for every cell, I want to assign the state that that cell falls into. There's a variety of ways you can do this in R, but I'll show you the way that I would do it. There's a function in the SF package called stjoin. So it's a spatial join. So I want to take the fold level trends in spatial format and join them to the states. And I'm going to, by default, it will keep any points that that are not in any state, but I don't want that. I want to remove those points. And to do that, you say left equals false. This is going to return a spatial object, but I'm actually done. I don't need any other spatial operations. So I'm going to also say st drop geometry. That converts it from a spatial data frame back to just a normal data frame. Let's look at it. Exact same data frame. The only difference is that now every every cell ID is associated with a state. So this cell is in Washington. So the next step is to do exactly the same thing I did for that previous simple example and calculate the abundance weighted trend within each state now. And I'm going to do that for every fold. So the result of this will be for every state, I'll have 100 estimates of the percent per year trend. I'm going to call this data frame trends, states, folds. I'm going to take the data frame that has the state attached. I'm going to group by both the state and the fold. And then I will summarize. And I'll do exactly what I did before. I'll say the percent per year trend at the state for the state and fold is the abundance weighted trend using that same formula. So the sum of the abundance times the percent per year trend divided by the sum of just the abundance. And I'm going to do a little thing to just remove the grouping. I grouped the data frame here. I don't need that grouping anymore, so I'm going to tell it to drop the groups. Okay. See what this looks like. All right, so for every state now, you'll see we're going to have 100 trend estimates. So for Arizona, the first fold, the trend is minus 0.9. The second fold, minus 3.89, and so on. And now because we have this set of 100 folds, that allows us to summarize across them, and we can calculate the median, which we already did before, but we can also calculate confidence limits. So we can calculate, in this case, I'm going to do the 10th and 90th percentile. Okay, I'm going to call this trends states. I'll take the data frame I just created. This time I'll group just by state, not by fold. And then I'm going to summarize, and I'll calculate the median value. Oops. I'll also calculate abundance, percent per year abundance, the lower confidence limit. And for that, I'm going to use the quantile function. To calculate the 10th percentile. And then I'll calculate the upper confidence limit. And in this case, I'll use again the quantile function, but this one will be the 90th. And again, I'll just drop the groups. Okay, whoops. Forgot to put state in here. I didn't actually group by anything. Okay. All right. So now we've gone from 100 folds for every state. We've collapsed that across the fold. Now we have one estimate for every state. The last bit here, it's just some ggplot code that will make a really simple map. It's joining those values back to the state polygons that we had and then creating a simple map. So this map is showing sage thrasher breeding percent per year trends within the states. And you can see here the sort of darkest red values are going to correspond to a trend of like about minus 5% per year. And then these light values are very close to zero, but a little bit on the negative side. And we could, for example, create some way to visualize also the uncertainty as well, because we have both the median and the uncertainty. Okay, I have one more fairly quick example. Tom and Andrew, are there any questions I should address now, or just get on to that example? Okay. Okay, so the last example, I'm sticking with sagebrush. In this case, let's say that you're interested in not the trend for one particular species, but the trend for some community of species. So, it could be grassland birds or deciduous forest birds or something like that. In this case, let's pick a set of 3 sagebrush species that we have trend estimates for. I'm actually just going to paste that in rather than type it out. So I have oops, 3 species here. Our friend, the sage thrasher, and then sagebrush sparrow and brewer sparrow. So we're interested in the community level trend for that group of species. We can start by just looking at the trends runs data frame for this set of species. This is a worthwhile thing to do just to make sure that the species are all for the same region and season. So for example, if you don't want to combine, a North American trend and a European trend or something like that, because it doesn't make sense. So just a quick check to make sure we're, in this case, all 3 are trends for North America. Now I'm going to download the trends for these 3 species. One cool thing about the trends side, because it's a much simpler data set, you can download multiple species at once. So I can give the eBirdSE download trends function, I can give it that full list of species, and it will download all 3. In this case, it's just telling me that I already pre-downloaded them. And similarly, the load trends function also works with multiple species. Since the data frame, it can just combine all 3 data frames together. So I'm going to call this thing trends sagebrush species. So it's that same load trends function, but rather than giving it a single species, I'll give it the full, that vector that has 3 species names in it. Let's look at it. It's exactly the same as before. The only difference is that rather than just having 1 species, so this is brewer's sparrow, but at the bottom of the data frame will be sage thrasher. And then the sagebrush sparrow will be in the middle. And all I'm going to do at this point is just calculate the cell-wise mean. So I'm going to take the mean % per year trend across the 3 species for each cell. So I'll call this trends sagebrush. So the data frame with all the trends is trends sagebrush species. In this case, I'm going to group by, remember that column, that SRD ID, that is the unique integer for each cell. I'm also going to throw in the latitude. I'm also going to group by the latitude and longitude because that'll come in handy for plotting later. And I will summarize across the 3 species. I want to keep track of, for every cell, how many species contributed to that cell, because the ranges of these 3 species don't perfectly overlap. So you might have some cells that have 1 species and some cells that might have all 3 species. So number of species, there's this function n that will just count the number of individual species contributing to the estimate. And I'm just going to take the mean trend across 3 species. And again, I'll drop the groups. Oops, misspelled summarize. Okay. So now we have, for each cell, we have an estimate of the community-wide % per year trend in relative abundance. And finally, I have a bit of code again to make a really simple map. I'm actually going to just remove the cells that don't have all 3 species and just look at the set of cells that have all 3 of these sagebrush species. I'm going to convert to a spatial format. And finally, I will make a really bad map. There's no context in this map, but you can imagine it's somewhere in the Western US. The blues indicate increasing trends and the reds indicate decreasing trends. that is it for me for the applications. I think we're going to transition to questions, but first, Andrew is going to speak about some maybe more advanced types of analyses you could do. And I'm going to share. Oops. Okay, you good to go, Andrew?
Yes, thanks, Matt. Just a second. Perfect. Well, yeah. So thanks for those examples. Those are really useful ways to use these spatially explicit trend estimates. And of course, the fact that the eBird trends are spatially explicit makes them a really powerful tool to understand what's going on with bird populations. And as part of this, we've seen a lot of interest in overlaying maps of habitat features with eBird trend maps to start looking for patterns. For example, are there correlations between a specific habitat type and population trends? So for example, here's a map on the screen that you're seeing showing the percent cover of dense herbaceous vegetation. This is a land cover type that goes into the models from remote sensing, and then a map of trends for brewer sparrow. So what are the things that we need to think carefully about when doing these sorts of comparisons? Next slide, please. So first and most importantly, overlaying habitat over trends gets you information about correlation, but not causation. This is true for any spatially explicit trend estimate, including these ones from eBird. But often correlations are useful to still gain a better understanding of the potential drivers of trends and perhaps prioritize areas where more study might be needed. Second, one thing that I think is really fantastic about these eBird trend models is that environmental information on habitats and habitat change is already baked into the trend model. So changes to habitats are reflected in these trends. This is different from many traditional models for survey-based data where trends might be based only on differences in counts between years without explicitly using habitat change for a trend estimation. So this means that coarse land cover variables, like the ones that you get from remote sensing or the ones that are already included in the models, those sorts of variables are good candidates for looking at correlations by overlaying habitat change with the trend estimates as well. So essentially, you can start to pull out habitat information and habitat inference that was already baked into the model. This is something called explainable or interpretable machine learning, where we're using machine learning methods to generate the trends using habitat change. And then post hoc, we can start to pull out information about how the habitats affected trends in the model. Last, it is possible to compare trends to custom variables that you might make yourself or might be available only for certain regions, but this requires some careful thought about the variables that you're using. Appropriate variables for this sort of exploration will have spatial resolution and temporal resolution that can be adequately captured by the trend model in the first place. So for example, if the custom variable that you want to use is radically different from the variables in the original trend model, like maybe it's very, very high resolution, like 30-meter resolution, or maybe the time periods don't quite match up, then you might not have the power to detect an effect of that particular covariate on these spatially explicit trends from eBird. Thanks, Matt. So if you've got questions about kind of habitat inference and habitat overlays with trends, please feel free to put them in the Q&A and we'll answer them. Matt, are you frozen right now? No.
So we've had a lot of questions about what we are estimating, and this is the slide that I wish I'd put in the beginning or part of my presentation. And there's an FAQ item about this, but and the font is a little small. That's better. We'll just zoom that in a little bit. You know, the blue line here would be what's actually happening with the population. This is just an example over a period of time. And the red line is what we're estimating from the start and the end. But notice also that the red line doesn't exactly overlap the start and the end of the blue lines. That's also not what we're estimating. We're not estimating 2022 versus 2012 specifically. That the trend is estimating the mean percent per year change over the whole time period. And so the model is seeing some of the interannual variation, and it is calculating a mean effect over the whole time period. And so that one line that we're calculating between the two time points is incorporating some of the interannual variability, but we're not making specific estimates of each year in what we're producing. But it is not exactly what's happening from between 2022 and 2012.
Great. So if you have any other questions that you'd like us to answer during live Q&A, we can stick around for longer. So please feel free to put those in the Q&A. We can answer them live, and we can be here until, was it 1:30 or 2?
We can be here till 2.
We can be here for up to another half an hour.
Oh, question live here. Someone's asking if there's any interest in the future to combine eBird data with field data to improve model estimates. Yes, we have a couple of staff working on integrated population models that combine eBird data, trends estimates with other sources of data. I believe some of that is field-based data.
Yes.
And there's a strong interest in data integration, which is kind of an ongoing research topic for us, but it will be a while before they're combined in any large scale way.
Yeah. Another question for you, Tom. How do you think about how to measure the performance of the models? We've seen R² come up a couple of different times. Do you consider other metrics like root mean squared error?
Yeah, Matt may be able to answer some of this a little bit better. There are a suite of performance metrics available in the summaries about the trends, and I know R² is one of them. There's a couple other ones, and I don't, again, Matt, maybe you can chime in with what ones are specifically in there, but there are some measures we at least use internally to evaluate how often we get the directionality right in those simulations, and basically gives us a sense of power. So we can look at that quality as well. We haven't used a root mean squared error to evaluate the quality, but if you also look at the paper, there should be some discussion about some of those understanding how often the directionality is wrong in the supplementary material there as well.
Yeah, I will say, like, the only thing that's available through the R package, like available publicly, is R², and then the beta naught, like the directional error. Those are the only two the only two metrics that you all have access to. So there's a question about someone saying they've been told it's not appropriate to compare relative abundance for one species in 1 year to basically you can't compare relative abundance between years. I think that's something we talked about in the last webinar. This is, I think, a question about the status data products, not the trends data products. Essentially, you know, we on a regular basis we update the status data products. So you might have an estimate for 2021 and then another one from 2022 for the same species. But there can be a lot of model changes between 2021 and 2022. So the changes you're seeing in those relative abundance values may have a lot more to do with different covariates we've added or changes to the model rather than actual changes in the population. So you never want to compare status data products between years. I don't know if maybe Andrew, you want to talk about ground-based confirmation of the estimates, like some of the Eagle work or something like that?
Absolutely. Yeah. So there's quite a bit of interest in kind of how do these things validate to on the ground data sets. And so this is a place of active research. We've worked, for example, with the U.S. Fish and Wildlife Service on validating some of these data products against the data that they have. I think a good example is work that we did with golden eagles. We've done this with bald eagles as well, where we compared estimates from eBird status to some broad scale survey data sets that they had done. For example, they ran 10 years or 11 years of aerial monitoring for golden eagles, where people were counting eagles from planes. That allowed them to estimate the actual abundance of golden eagles at the level of these flight transects that they were flying in the planes. We could compare that to eBird status to see how well they lined up. And we found that these things line up quite well. If you were to model one versus the other, for example, you'd get a really strong correlation. Places where, and then from there, you can take that relationship between survey data and the eBird relative abundance to kind of rescale eBird to match the units of the modeled survey data to kind of get better ideas of the actual density of birds rather than just the relative abundance. When we do that, we find that those density estimates also overlap well with survey-based density estimates. So in the cases where we have done validation, we tend to see quite a lot of overlap, and it's been really encouraging so far.
So
Yeah,
there's
go ahead.
specific years, and unfortunately, no, we have the mean trend over the period of 2012 to 2022 right now. We are working on being able to extract specific year differences in the future.
And then there's this question about sort of inclusion of kind of legacy handwritten bird logs in these models. I mean, if the data can be put into the form of like a complete eBird checklist, and you have all that information like about effort and all that, date, time, location, effort, you could enter it as an eBird checklist, a historical eBird checklist, and then it would make it into our models. But that's at the moment, that's sort of the only way that sort of data would make it into the modeling. Oh, and then they're asking, will I be able to upload the entire data set, or do I have to do one checklist at a time? There is a sort of a bulk upload functionality where you can put the data. I've actually never used it myself, so I can't comment too much on it, but you can put the data in a specific CSV or like a tabular format and upload all at once, which would avoid you having to individually create a bunch of eBird checklists one at a time. Seabirds. Do we have any seabird trends, Tom? I can't actually remember.
I don't believe we do, unless it's like a very like coastal species.
Yeah.
Yeah, basically, you know, there's very limited data for seabirds. One of the other things is the primary covariates that we have for seabirds right now are subannual covariates. They're monthly specific through time, things like sea surface temperature and sea surface temperature anomaly. And in our modeling framework, it's very complicated to try to understand how those things change through time as well. So I don't foresee seabird trends in the too near future. Don't need it.
Ooh, question about fun upcoming improvements.
Upcoming improvements. Well, the primary, we're doing a number of research threads focusing on improving the trends generally. One of those is to handle more regional scale effects, and basically, yeah, improve the quality of the estimates regionally within a species range. That's one thread. We're also continuing to do some validation work on the trends to kind of highlight their reliability. So that's going on. And then there's work about some of the interpretability of the trends related to, well, Andrew, you're you're leading that work.
Yeah, we are working on different ways to kind of glean inference on how habitat and environmental change impacts bird population trends. Both post hoc methods that use these modeled outputs as starting points, and then within model applications where we try to like get that information directly from the model. And that's an active area of kind of quantitative stats research.
And I would say that, yeah, we're also trying to get to those year-specific estimates as well.
Maybe just like taking a huge step back, like when the new status data products are released early next year, I think probably the main website sort of public facing change, this is not a trends thing, but on the status side, is that you will be able to get continental proportions of populations. So for example, if you select currently for say a globally distributed species like barn swallow, if you select the US, you'll be able to get, you can currently get the proportion of the global population within the US, but as of the next release, you'll be able to get the proportion of the North American population that's within the US.
That'll be a nice update that makes proportion of population metrics more comparable between species that have global distributions like barn swallow or golden eagle and species that are more localized and endemic to a single continent. Questions are kind of dying down, so we'll give it one more minute. If you have more questions, please feel free to put them in the chat and we'll answer them live, or in the Q&A and we'll answer them live. And if there's no more questions after about a minute, we'll sign off.
I'm just going to post my email address in the chat. If you have any sort of like R related issues with any of the material from today or the other two sessions, send me a message. And I really want to make sure everyone's able to go through this material without running into any bugs or any hurdles.
Okay, we've got a couple of questions popped up. One is asking about the reliability of snow hail data in Canada during the summer. There's two ways to evaluate the veracity of the data that we have. One is that in the species table about the data products, the expert review assigns quality scores for each season. And I'm sure for snowy owl, it's not very high because there's such low data in the Canadian Arctic, and the estimates are less reliable there. And then we did briefly discuss, I think in the last webinar, how there are predictive performance metrics available in the R package. And we haven't provided a ton of guidance on how to use those, but if you're familiar with modeling, there you can look at that, although it sounds like this user maybe is not. So at least for now, the starting place would be to look and see what quality score each season has been given. And if it's not a 3, which is the highest quality, then maybe don't use it.
Great. There's a question in the Q&A. Are there current collaborations with conservation organizations that are using eBird data? And the answer is a resounding yes. There are dozens of ongoing collaborations between conservation groups, both within and outside of the United States, that are using eBird status and eBird trends. So for example, I work very closely with a number of different state agencies with a few dozen joint ventures, and then also federal agencies in the US, like the US Forest Service or the US Fish and Wildlife Service. So if you're interested in kind of learning more about those ongoing different collaborations, or if there's applications that you'd like to work together on, please feel free to reach out to me. I'll put my email in the chat as well. All right, looks like we're all set with questions. So thank you so much, everyone, for joining for these 3 different sessions. I hope these were informative and enjoyable. We definitely enjoyed putting them on and having such great attendance from the audience. So thank you so much for kind of joining along with us and going through these eBird applications with us. And Matt has his email in the chat, so feel free to reach out to us or our team if you've got specific questions. And happy coding, everyone.
Thanks for joining. Take care.